Just gonna wait to see who all we can get. I don't think it'll be a lot of people. What are you doing? Hopefully people will at least join late. We'll do it with or without them, but I was kind of hoping for more people than this. My hopes are always really high at the beginning of the semester. Meatball, stop it. Okay. Anybody have anything exciting for dinner? I ate like a whole container of hummus. And then I was like, maybe I shouldn't eat dinner. That was like a lot. But I had it with veggies, so that makes it okay, right? Well, we'll probably get a couple people kind of wandering in as we get going. Whatever, we're just gonna go ahead and get started. All right, I put like a PowerPoint together for this. I put a lot of PowerPoints together for like no reason, probably don't need a PowerPoint for things, but we have a PowerPoint for this. So yeah, so we're gonna go kind of over unit 1. I'm gonna just basically run through the study sheet. That's really all I'm doing. And I'm not gonna like go into a lot of detail or really explain a lot. I mean, that's what the lectures were for, right? So I am going to touch on some stuff that I feel like people forget about, or some stuff that I think is worth focusing on. I'm not guaranteeing they were on the test. I don't remember everything that was on the test, but to me, in my mind, they are worth focusing on. So, probably means that I felt that way when I was writing the test too. So starting with chapter 1, microorganisms need a microscope to be seen. That's just the definition of what a microorganism is. And so you can't see them with the naked eye. Viruses and prions are acellular. That means that they are things that can cause disease in your body that technically are not made of true cells. What is a cell? A cell is something that is membrane bound around the whole, like it's contained by a membrane and contains genetic information that encodes everything that needs to be, and it can divide on its own without necessarily requiring stealing host machinery or something like that, like we see with the viruses, right? So that's the difference there. That's what I mean by acellular. What is happening around me? I don't even know. The eukaryote, A being without, karyo being nucleus. So eukaryotes do not have a nucleus. Yeah, I don't know what else to say. And because of that, I mean, think of like if as life would evolve on the planet, of course, it would be the simpler things that would evolve first, and then the more complicated things. So let me get this bar over here. I wish there was a way. There's a way to make it go long ways, but I don't remember how to do it. It's smart enough. Oh well, whatever. Just leave it over there. Okay, whatever. Most of Earth's atmosphere was made by microorganisms. We're talking here about cyanobacteria that perform a photosynthesis on the membranes of their own cells, their inner membranes. And so done with that, and we're talking about like the plankton, the phytoplankton. So phyto is just like photosynthesis, just think of it that way. But those guys that are green, they're going to make most of the oxygen. There's just more of them doing it cell per cell than the plants, and they've been around for so much longer. So that's what we're talking about, even since before plants they've been doing this. They're the ones that changed the atmosphere originally and then made it so that plants and other animals could even exist. Well, first plants and then other animals could exist. So pretty cool stuff. Germ theory says that communicable diseases are caused by pathogenic organisms, and they don't just spontaneously generate. There was a thought back in the day about, diseases were caused by these, like miasma, which would be bad smells. So just the smell itself could lead to a disease. There's the idea of spontaneous generation, which meant that, even like maggots or something would just appear on meat as it got old, and not necessarily have anything to do with flies laying eggs on it or anything like that, because they couldn't see it, they didn't know. So that's what spontaneous generation was. And Pasteur was like, you guys are just full of a whole bunch of honky. That's not what's going on. And he proved it with his experiments that you guys don't know the details of, you don't need to. And then Koch's postulates are used to kind of verify causative agents. And so Koch was the one that discovered that Bacillus anthracis was the cause of anthrax. So he used that on his own method. Nowadays we use Koch's method, but we just use it with molecular means and not just, old school. Or they inject it with you isolate something, like, I think this is the cause, you put it in a mouse, it has the same symptoms as the people, then you take it out, whatever you had put in the mouse had made it sick, you re-isolate it again, it's the same thing. That's what Koch's postulates was. So, but yeah. I think there's a lot going on. There is. It's a lot more than you guys can see happening behind me. There's a little dog sitting, you can't tell because he's black, but that's a little dog right there. That's Spaghetti. And Meatball is migrating around the room. Meatball probably has some sort of like Cushing's disease or something. He always is trying to eat, always, even when there's not food, he's always looking for it. So I think he probably has Cushing's or something, but we just monitor how much food he eats. He's usually okay, but he's a little chonky. But he's wandering around looking for food right now. I have 3 dogs in here. There's another dog over here just chilling, and that's Ripley, but she's like 13, so of course she's chilling. But yeah, that's what's going on, and I keep hearing them move around. It's like, what are you doing? So moving on. Aseptic technique, we learned about this in the lab as well. Aseptic technique is just meant to prevent contamination, and this is applied in all sorts of aspects of your life. It's not just in studying microbes, right? Whenever you wash your hands, that's aseptic technique. Whenever you are, even taking a shower, or I don't know, all sorts of stuff. So, anytime that you're washing anything, preventing any type of contamination anywhere, dirt or anything like that, that's technically going to be applying to aseptic technique. So preventing contamination one way or the other. All right, taxonomy is stupid. but you have to learn it. So it's one of those things that's going to show up in your chapter 1 or in your unit 1 exam. I guess for you guys also unit 2, because it depends, right? We'll see what your average is like for unit 1. And if in unit 2 you want to take, I don't know, it has to be voted for the whole class. So we'll have to figure it out that way. But we'll see what it looks like. Hopefully you do great and you won't need to, fish for better grades. But yeah. Taxonomy, though, it's stupid. I do have this the only times that you're going to see it, maybe on the final. Domain is the big one, and most of you guys were writing about this in your questions. I do read your questions, by the way, for the extra credits. And I just read them. You guys are doing a pretty good job for the most part of writing your own questions and not copying too much either from each other or from ChatGPT or something like that. At least if you are, then you're doing a good job of hiding it. And if you're like, oh, I am using ChatGPT to write it, and then you're rewriting it in your own words, that's kind of the point. So I get it though. So yeah, so taxonomy, domain, most of you guys know this one: bacteria, archaea, eukarya. So there's 3. If you can remember eukarya and the bacteria, which we're learning about in the lab, right? Then archaea is the only one that's missing there. So you just got to remember that it's the older one, archaic, archaea. Okay. And it kind of falls in between the two. We weren't really going to talk about the archaea too much. They're just the extremophiles. They're the ones that live in extreme environments. So the boiling hot water, high acidity, and stuff like that. So, but whatever. After domain, you could take like eukarya, which is like where we belong, right? Because we're not bacteria or archaea. So take eukarya, you would divide that up into kingdom, phylum, class, order, family, genus, and species. So that's the order. Whatever you need to do to remember that order, be aware of it. Then we have the binomial nomenclature. It's a 2-name way to refer to organisms. It's the genus and the specific epithet. And when I'm talking about binomial nomenclature and the genus and the specific epithet, there's a specific way that you're supposed to be writing them. And I'm going to talk about it in the next slide. So yeah, aseptic technique is everything. Anytime you're trying to control spread of microbes, using Lysol on your countertop after you made chicken or something like that is aseptic technique. So know the taxons in order well enough to know if they're out of order, right? So if I say, does order come before or after kingdom, then you should be able to tell me, yeah, yes, true or false, basically. Capitalized genus, lowercase epithet is how it's always going to be. So think of E. coli, right? Or S. aureus. So the first word is always going to be capitalized. Escherichia or E, even if it's abbreviated, it's going to be capitalized, right? And then the second word, the specific epithet, is going to be lowercase. So coli. And if you're typing it, it'll be italicized. And then if it is handwritten, it'll be underlined. So I'm like 99% sure there's going to be a question about that on the test where it's going to be like which organism name is written correctly. And I think there was one on the quiz as well that was similar. Anyways, which one is written correctly, and one will be capital, and then one will be capital and little, and some will be italicized, some won't be able to know what it's supposed to look like. I feel like remembering E. coli helps a lot because most people know that E. coli is big E, little coli, but I don't know. If you don't, then I'm not helping you at all. All right, chapter 3 kind of ties in with chapter 1. It's our basics. So we have our 5 I's, and they are in an order, and I don't need you to necessarily remember the order because what I'm going to ask you about is sort of like what you had on the quiz, which is like which of the following choices doesn't belong in the 5 I's or something like that, or what I is missing, and I'll give you 4 I's, and then you have to tell me which one isn't in the list. So that's more likely what the test question is, I'm pretty sure. So inoculation, incubation, isolation, inspection, and identification. be familiar with those words, be able to see them on paper and know that those are the correct words that need to be in there and know that other words are not. So different types of media, selective and differential. Selective only grows certain kinds of bacteria. Other things won't grow on it. Differential, it will change color, usually in response to something the bacteria makes whenever it's breaking down a product, some sort of, I want to say ingredient in the media. So if it's breaking down a sugar and it's making acid from fermenting that sugar, then that acid can change the pH, and a pH indicator can change its color as a result. We'll see a lot of that in the second part of the lab. Quite a lot of it, actually. But yeah, so that's a pretty common one, is pH change. And then we have, but technically, that differential aspect, that pH change, that color, that itself doesn't inhibit anything from growing, right? So if you're looking at differential media, that itself isn't going to be the thing that keeps something from growing or favors something to grow. That's the selective part. And yes, you can have both at the same time. So media can be like McConkey agar, can be selective and differential. So we'll be getting into those definitely in the second part of the lab. Enriched media, that's going to be media that has growth factors in it. And a growth factor is something that an organism requires in order to grow. Bacteria could need any number of things. It just depends on the strain that we're talking about, the species that we're talking about. But in humans, you need vitamin C, right? If you don't get vitamin C, then you get scurvy. You get B12, usually from eating meat, but if you're not a big meat eater, or you have a problem absorbing it in your meals, you might need to get shots or do like supplements or something like that. So, but if you don't have them, you will do very, very poorly and could even die from a lack of those. So those are growth factors to us. Bacteria, other microorganisms have similar things. Could be vitamins, could be other things. It's not always vitamins. Anytime that a bacteria requires something, anytime that an organism requires something, a growth factor, in order to be supplemented into their diet in order to live. Why did my monitor bounces so much? That is a fastidious organism. So humans are fastidious. So fastidious bacteria need their growth factor added into their media. So they only grow on enriched media that has been enriched with their growth factor. So there's ways to look at that. All right, looking at microscopes, magnification occurs because of bending of light by lenses. Refraction is light scattering. Objective lenses will form the real image of the ones that are closest to the specimen, right? So we've already seen our microscopes. We know that when the light comes through the specimen on that glass into that, then it is going to hit the objective lens first, the one that's on the little rotating nose piece, right? So that objective lens, we'll see it first. So that's the real image, the image of the actual specimen. It hits some mirrors and goes through another lens in the eyepiece. That's the virtual image in the eyepiece, okay? If you're trying to calculate total magnifying power, here we're talking about the objective lens that you're using, the ones that will rotate between, and the ocular lens, which is usually, you don't change that. So ours are always at 10x for our ocular lenses, which by the way, you have in microscopes. John went and got them for me and put them all in the lab. So I'm incredibly thankful for that. So incredibly thankful. And I've got the cultures growing. So I'm just so excited to actually have things work. It just was one of those, like just the timing. It was like the perfect storm. But so we've got microscopes. I'm really excited. But yes, so the magnifying power, if you're trying to figure it out from a microscope, is going to be the objective lens times the ocular lens, whatever objective lens that you're using. Oil immersion is going to prevent light scatter because oil has same refractive properties as glass. So instead of it going from the glass, the light through the glass, and then through the air and then back into glass, now the oil kind of makes it seamless. So it prevents light scatter. Heat fixing kills cells, secures them to the slide, and preserves cellular components. So that whenever you stain them, it's ideal for those situations where you don't need the organism to be alive. So that's pretty useful there. Staining provides contrast, and there's different kinds of stains. yeah, I don't know what else to say about this. Yeah, negative stains and positive stains. I'm just trying to think about this, but yeah, you could think of negative as being acidic typically, or the basic being the positive. And those be attracted to cells according to the charge on the cells. So your cell surfaces have a negative charge to them. Think of those phospholipids. They're on the surface of cells. They have a negative phosphates. If you hear phosphate at any point in this entire course, please remember, if you remember anything about a phosphate, that it is negatively charged, okay? Because pretty much everything in this course is going to be driven by the fact that phosphates have a negative charge. We're going to talk about energy that the cell gets from metabolism and ATP production, which is our energy molecule. And the ATP is adenosine triphosphate. Oh my gosh, 3 phosphates, 3 negatives. If you know anything about like a North Pole to a North Pole magnet, they're going to push each other apart. That chemical aspect, that I guess you could even say physics, it's like atomic physics. But that aspect of those phosphates pushing each other apart, but still being bonded by those covalent bonds, that's what stores all the energy. It doesn't want to stay like that. It's like a spring just waiting to be smacked back together, or you know, just trying to shoot that other magnet away so that it doesn't have to be near it anymore. All that energy that builds up in those bonds. So super important, I would say, phosphates, negatives. That'll help you through a lot of this as we go through, especially in Unit 2. Because there's a lot of chemistry involved, and you wouldn't think it, but we talk about DNA replication, we're going to talk about metabolism. It's all going to be a ton of chemistry, so just bear with me. So, but I love those chapters. They're some of my most favorite chapters. So, yeah, and that means I get excited. Though I'm not saying that means that you need to be worried. It means that I love explaining it. So, yeah. Anyways, phosphates. Negatively charged on the surface of your cell because, phospholipids, and those negatively charged phosphates on the surface of your cell, those phosphate heads sticking out on the outside, negative charge. If you put a negatively charged stain onto that, a negative stain, it's not going to stain the cell, right? It's going to repel it. Negative to negative, it's like North Pole to North Pole. They don't like each other, right? Opposites attract, not samesies. So the negative stain that's coming in on that negative phosphate head, they are not going to want to actually be truly associated with one another. So you're going to stain the background instead. With the stains that are positively charged, those positive stains will get associated with the negative phosphate heads, and they'll actually become one and bind together and stain the cells themselves. And that's how you get that true staining factor that we see with our cells, like we did with the Gram stain today. So that's how stains kind of operate in general. And that's definitely how simple staining works. Well, are you growling at me? I'm pretty sure he did. I'm pretty sure he growled at me. He doesn't like it. Sometimes he complains if he's not getting petted. So it's just easier to watch them if they're all in the small space in the office with me. Yeah. So differential stains, like we did with the Gram stain, those differential stains are going to tell you differences between cells. So whether they're Gram positive or Gram negative, whether they are acid fast or not. If it is asking any question between a positive, yes, it is this, or a negative, no, it isn't that, then that is a differential stain. So obviously, whenever you're staining with a simple stain, it's not differential. You're just seeing if cells are there. But if you're trying to tell the difference between the cells, differential stains. So that includes Gram stains, acid-fast stains, capsule stains, endospore stains. We'll be doing all of those in the lab. Okay, so with the 5 eyes, remember, pick out the imposter eye, that you need to know it that well. If I gave you a list of 5, or 5, if I put one of them's fake, you need to know which one it was. Or if I put 6, which one doesn't belong, you know what I mean. That sort of stuff. What is the difference between selective versus differential? So be sure you know the difference between them. Can they be both? I just told you they can. You have something in there to prevent one thing growing or another. Guess what? You can have stuff in media that can prevent Gram positives from growing. And that would be favoring gram negatives. You don't need to worry about what it is right now, but there is stuff you can add to media to keep gram positives from growing. Go figure. It has to do with preventing probably their thick peptidoglycan walls. Yeah. So the gram negatives will grow on there. That'd be preferred. And then there might also be a color changing factor of whether they can use lactose or not, which is exactly how both EMB agar or McConkey agar would work. And we'll be way more familiar with them than we want to be later on. But yes, you can absolutely be both. Okay. Know the difference between them and what they mean, selective, differential. Be able to delineate the differences and relationship between enriched fastidious organisms, growth factor. If I just give you the word enriched, fastidious, growth factor, you need to know which one refers to the media, to the organism, and to the thing added into the media that the organism needs. Okay. So think about it that way. So you can understand those terms better. Immersion oil reduces refraction. I always say, like you've seen me say it in a whole bunch of things, that immersion oil, when we use it on the 100X in the microscopes, which a few of you got to see me actually do that today. And we will be doing that next lab since we have our microscopes and yeah, you guys will be actually using them. And I think it's good that we got to kind of go one by one through the microscope and I got to see you guys at least get familiar with that 4X. So we kind of all knew where we were at there. I think that's actually a good thing. So, so I usually don't get to give that much attention when everybody's all at once. So it's kind of hard then. Anyways, so I always say immersion oil reduces light scatter, but guess what light scatter is? Refraction. That's the same term. That's all this kind of like just pointing you in that direction of refraction is light scatter. Just try to use the word interchangeably when you're talking about microscopy so that you can be more familiar with it. And so that if you have questions about that sort of stuff, you know that it's the same, it won't be a problem, it won't be as daunting. Real image versus virtual, which one occurs where in the microscope? Remember, real image is one that hits the lens first. So the first lens that we hit is the objective lens. And so that objective lens that's like turning around on the little nose piece, that will then, the light will go through that. Real image there. Then mirrors, I don't know how many there are, probably like a million more than I think there are. And then through the ocular lens, again, another magnification step, 10x, right? That's going to create that virtual image that you'll see. Is differential media like differential stains? Yes, it is actually. So anytime you hear the term differential in any of microbiology, just think that it tells the difference. I mean, differential, don't get overwhelmed by the term. It literally just means different, like telling the difference. So differential media, telling the difference between whether it can do, whether it metabolizes this or not, whether it turns the media pink or not, basically. But not preventing anything from growing or not. It's just telling you whether it does a thing or not. Differential stains, we're looking at whether it's gram positive or not. Is it acid fast or not? Basically, that's kind of how it goes. So whether it has a trait or not, okay? They both do that. All right, into chapter 2, we have chemistry, types of bonds. A lot of you guys are really familiar with this, so hopefully this is just a refresher. Covalent is sharing electrons, ionic, transferring electrons. Remember, that's going to be like sodium chloride, sodium ions and chloride ions, most chlorine ions, either way you want to look at it. But yeah, we know of those as being ions. You guys are probably familiar with those being ions. So if you can remember sodium and chloride, those are ions. They make ionic bonds. Yes, those absolutely do go together, believe it or not. That's not trying to bumble jumble up all those terms in your head. Ionic bonds can lead to ions forming because one is totally completely taking the electron away from the other one. And basically like, sodium being an Na plus and chlorine atom being Cl negative. It's negative because it stole the electron from sodium. Sodium is positive because it got its electron stolen by chlorine. So that's why they have the charges that they do. They are ionic bonds that are formed between sodium and chloride. That will also bring us to being able to explain how redox reactions go when we get to redox reactions in Unit 2, because we will be getting back to those. I'm sure if you guys remember redox reactions at all from chemistry, you're just thrilled to get back into that. But yeah, just electron exchange is all, okay? Hydrogen bonds do not involve exchanging or sharing electrons. The actual electron of hydrogen and whatever other atom that it is like just associating with, that's the bond that occurs with them isn't due to electrons between those atoms at all. What happens is the hydrogen, because hydrogens are very much not electronegative, hydrogens will not hold on to their electron very well in their covalent bond to their neighbor. A lot of times it's an oxygen. So think of H2O, right? If you can remember that H2O, the O has slightly negative and the H's have positives, that pretty much spells out how that electronegativity works. And whenever those slightly positive hydrogens come around a slightly negative oxygen that's in its vicinity, then they can have an association with one another called a hydrogen bond. They are weak bonds. There's no actual electron exchange. Most of your macromolecules in your body are largely, their actual shape, which is their functional aspect, largely held together by hydrogen bonds. Your entire DNA, listen, your whole DNA is 2 strands held together only by hydrogen bonds in the middle. There's no real bonds in there. Everything about your proteins, like you have your proteins and they're like amino acids just in a long straight line, right? Well, how like this guy is like, you know, attracted to this guy under here, how it starts to fold like that. And that can lead to formations of a helix. That's the secondary structure, by the way. So amino acids, primary, how they start to interact, that's the secondary. And then how those helixes interact with the beta sheets across the way, or the helixes across the way, or they come together from across in the same polypeptide. And I don't know, this looks ridiculous, but it's basically how it is. That's the tertiary. And once you have that blob basically created from all these attractions, that's all, like, not all, there's a few disulfide bonds in there. Yes, I know there are, but it's largely hydrogen bonds causing that folding to happen the way that they do. So what happens whenever you expose, let's say, a protein that is largely held together by hydrogen bonds to acid, which is very, very, very high in hydrogens, right? H pluses. That's going to disturb the ability of this being attracted to this. It's no longer as attractive because there's so many other hydrogens, it's not even noticing that anymore. So now your protein becomes unfolded, or what we call denatured. These things are so important in biology. I can't even begin to stress it, how important it is. So understanding protein structure is very key in getting through this course as well, I feel like, because we're going to talk about it in almost every single chapter. So that comes back to understanding organic chemistry, covalent bonds, ionic bonds versus hydrogen bonds, okay? So cool. So organic molecules, carbon, hydrogen backbone. It's not just carbon by itself. CO2, carbon dioxide, is not technically an organic molecule, inorganic, because it doesn't have any hydrogen associated with its backbone. Polar molecules have a charge. Non-polar molecules are totally neutral. So that leads us into understanding a little bit better about hydrophobic and hydrophilic and amphipathic. Hydrophobic, water-fearing, things that like oil, so fats. So we know oil is like very largely made up of lipids or fats. So fats are a great example of hydrophobic. Hydrophilic means water loving. Anything that mixes well with water, like if you mix vinegar with water, it just mixes, right? If you mix alcohol with water, obviously it mixes, because we have like 70% ethanol that you can just, or isopropyl alcohol or whatever, that you can just buy. If you go to get a cocktail at a bar, it's usually mixed with water. We know alcohol mixes well with water. That means it must be hydrophilic, water loving. So anything that mixes well with water, water loving. So they must have a polar aspect to them. If you know anything about the structure of molecules, then, or those R groups, those R chains, the OH group on alcohols. I'm just now seeing this chat. I'm sorry. I'm so enthralled in my own discussion and words. I'm not even seeing this. Our tertiary structure, proteins considered functional, they can be. So it just depends on the protein. So once you get all those big old globs, so you get your secondary, you get your alpha helices or whatever, and then like continue, just pretend like this is the continuing rest of this. So we have alpha helix and alpha helix, and they're attracted to one another and making up that big old conglomerate of the tertiary of that one single polypeptide. It can, in some cases, be functional. A lot of times, once we get that big old conglomerate made, we're like, yeah, they have to go find a buddy. It might be the same kind of buddy, or it might be some other molecule altogether. But a lot of times we need other things to bind with us to make us functional. So if this was our functional protein, our tertiary structure here, and we needed another buddy to come in and they come and bind together like that, and now it is functional. I mean, I don't know how it would work, but it just depends on the protein, right? So that's the quaternary. So not all of them really need to go into quaternary if they are functional at tertiary, but most of them will. Most of them are going to be multi polypeptide. So this glob and this glob come together to make a functional glob, a whole unit glob. And there can be many parts to some of these guys. They can have like just a whole slew of globs coming together to make their functional aspect. Good question. So amphipathic are phospholipids are the best example of that. If you can remember any of them, it'll be the phospholipids, right? We already know phospholipids. We just talked about how phosphates had a negative charge. That's polar, right? We know polar is water associating, right? Negative or positive. Positive will like the oxygens, negative will like the hydrogens. So it goes with water really well as long as it has a charge there. That lipid aspect, the phospho, and then the lipid tail. The lipid is just like a lipid. It does not like water, so it's hydrophobic. So that they are amphipathic. They've got a hydrophobic tail and a hydrophilic head. And the heads will all like bond together with their little phosphate negative, all their little phosphate charge groups. And just a little more complicated, of course, than that, but to hide the tails from the watery environment of your body. So that's what creates your lipid bilayer, because those phosphates that are charged will all conglomerate together on the surface, hiding the hydrophobic tails. And so they're on the inside. Other ones are on the inside like this. So they're creating two layers of lipids, and then on the outside, two layers of phosphate groups. That's the lipid bilayer. Anytime you guys are talking about, anytime you hear me talking about a membrane, that's the bilayer, the lipid bilayer. Always. Okay. Water is the most important solvent of life, of course. pH refers to the inverse log of the hydrogen ion concentration in a solution. Great, isn't that wonderful? We know that pH 7 is neutral, less than that is acid, higher than that is base. So if you can remember that, there's a rule that's great to follow right there, but I would remember more than that. If your solution has a pH of 12 and my solution has a pH of 2, which one has more hydrogen ions? And if you guys aren't sure about that, then it helps to remember how the pH scale works. The lower the pH number, like 2, mine would be the 2 and yours is the 12, right? So lower the pH, the more hydrogens. The higher the pH, the less hydrogens, because it is an inverse log, okay? That inverse is what makes that rule for you. I'm not going to make you go through 10 to the negative 7, and that means pH of 7, which is what it means. But that's basically how that's going to be working. Okay, so remember it is 0 to 14, that's the whole scale. 7 is neutral, halfway. Less than 7 is acid. Greater than 7 is base. And opposite for kind of the hydrogen concentrations. Hydrogen, 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 hydrogen. Lots of hydrogen, that's going to be your acid. Okay. Okay, where are you guys going? You leaving? You think you're leaving through that closed door? Biological macromolecules, carbohydrates, nucleic acids, lipids, and proteins. No, I would be very familiar with what they're made of. So carbohydrate is repeat. Hey. Stop. It's a painting fell. Yeah, I didn't hit it, but it just happened to fall. Anyways, come here. Okay, so be quiet. Really like not ideal. I don't know the subunits are for each of the macromolecules. So know that carbohydrates are sugars. I'm not really going to talk too much about saccharides or saccharides, but carbohydrates are made of sugars. I would be familiar with. Nucleic acids are made of nucleotides. So anytime DNA, deoxyribonucleic acid, so nucleic acid, nucleotides. So that's pretty straightforward. ATP, that energy molecule, falls in that category. It's an energy molecule, but it's still a nucleic acid. Okay. Lipids, oils and fats, cholesterol. Just remember, I would definitely remember that cholesterol falls into that. And when we talk about mycolic acid for a mycobacteria, that's a waxy thing that makes it kind of hydrophobic and hard to work with, makes it really like a chunky sort of aspect to it. Really, I don't know how to describe it. It's kind of looks dry and flaky and chunky when you grow it on a slant or something. So, but yeah. Yes, I'm sorry, proteins, amino acids. So if I were to say amino acids are the subunit of what macromolecule, you better be able to tell me that is protein, like that, especially because we're going to be talking about protein so much. Definitely be familiar with it. Okay. You can go away if you want to the screen. It's okay. It's just like adamant that it doesn't want to go away. Okay. Primary, that's just the amino acid. So remember, you have to start in primary school. Primary, it's the most basic one, just the amino acid sequence. Okay, like the ribbon. So you have a long ribbon made up of amino acids. Secondary, now those amino acids are starting to interact with one another. Like I was saying, they're curling over because they're attracted to one another, maybe creating a helix, like an alpha helix. Or if they're kind of repelled by their surroundings, a lot of times is what we see with those beta pleated sheets, and they'll like fold back in on each other, like, and then like back up the other way. And that's a beta pleated sheet. Those are some basic structures for the secondary structure that we see that is basically governed almost entirely by those R groups on the nearby amino acids. First, those nearby ones, when they start interacting like across the way from one another, then, like I said, you got this helix and then this helix on another part of the same polypeptide, and they start interacting with one another, that causing the big folds, the big interactions, that's tertiary. And once we have polypeptides, polypeptide is just amino acids linked together, it's that ribbon, as long as they're on the same ribbon, okay? Once we get away from one ribbon, and start incorporating this ribbon with this ribbon that's also been, and then they'll interact with one another. That's quaternary. That'll be the functional protein there for the most part. Like I said, sometimes you can have functional whenever it's tertiary, but that's not very common. So just reminding you guys, sharing electrons versus transferring electrons, which bond is which. So sharing electrons, that's going to be the covalent. We're going just back and forth between those atoms. But if we're just completely donating that electron to the other atom, that's ionic. It's going to create ions. And it says, what are electrons doing in the hydrogen bonds? Between the hydrogen that's attracted to the thing across the way, creating the actual hydrogen bond, there's no electron exchange. Dude, why are you barking so much? Ew. Right, so there's no electron exchange going on between the hydrogen bonds, but there will be between like hogging of electrons here, causing this hydrogen to be positive, which is positive. This oxygen out here is negative because it's hogging the electrons from the hydrogen back there, and so they're getting attracted to one another. Negative, positive, positive, and negative opposites attract. So that's how that works. But the actual hydrogen bonds themselves do not have electron exchange. So keep that in mind. They're weak bonds, no electron action. Hydrophobic, hydrophilic, and amphipathic, we kind of went through that pretty well. I feel like I don't really need to go back on that. What's an example of amphipathic? We talked about the phospholipids, how the pH scale works, that an inverse log, be aware of it, right? I'm not going to ask for any math equations from you, but if I ask which one has more hydrogens and then which one is more acidic, we're going to give you pHs on those. Know what those terms are in relation to the actual pH scale. That would be helpful. In a biological subunit, amino acids, for example, can you give the macromolecule, can you name the macromolecule that they comprise? So if I tell them, hey, what do amino acids make up, like I said before, and you should be able to tell me that it's proteins. Again, it's all multiple choice, so you can pick from stuff. And then protein structure levels, which I feel like we've gone over fairly decently at this point. Okay, chapter 4. It is the longest one, and I feel like there's a lot to understand of what's going on with the bacteria. It's a lot of terms, though, so that's the good thing about it. Just kind of memorizing the terms here. Bacteria and archaea, they're single-celled. They have an F1, like the base, right? The cytoplasmic membrane. That's the membrane that holds everything in. They at least have that. They have genetic material, but that's not contained within anything. It's just chilling in a region called the nucleoid region. Be aware of the cell shapes. Cocci is round, bacilli is rod, pleomorphic, no shape really to it. It's just rando. And then cell arrangements. Staphylo is irregular, strepto in chains, palisades like fence posts, that's rods. Remember staphylo, the grape-like clusters, really only applies to cocci, okay? And palisades really only applies to the rods. And then tetrads and sarcina, another thing that really only applies to the cocci. But tetrads is in 4s and sarcina is cubes. Capsules and slime layers. Capsules and slime layers are formed by the glycocalyx. Glyco should always trigger sugar in your mind. So glycocalyx, we're talking about the sugary coating. It can be loosely associated, which is the slime layer, or it can be tightly associated with which is the capsule. The capsules are important for adhering to surfaces, evading the immune system, and forming biofilms. Capsules are going to come up over and over again, so it's a good one to understand. Flagella are used for motility. It's the only appendage that bacteria use for motility. It is the only one. So bacteria only can get around if they have at least one flagellum. And the pili are for channeling DNA between, they're called sex pili. Fimbriae are for attachment. So those are all of the appendages that they can have that we're concerned about. I think there's like nano something wires or something like that. I'm not concerned about that. Flagellar arrangements. So this is what our terms are associated with that. Monotrichous, one at one end. Easy, right? Lopho, I don't know why lopho sounds like a tuft to me, but it does. Like the word sounds like a tuft, or I think of like lupa, like wolves, and lopho sounds like that, and they're fluffy, you know. So a tuft of flagella, so a bunch of them at one end. E. coli exhibits this trait. Amphitrichus, you have flagella at both poles, one at each end. Amphi, like amphibians, they can do both air and water. Amphi. Peritrichus, flagella dispersed all over the cell. I don't really know how to break that one down for you. Peri, I don't know what that prefix means. So I'm not going to look it up. I know trichus is like hair, but I don't know about peri, so it probably means all over. Anyways, cell envelope, that refers to the wall and any membranes and walls, whatever's covering that cell, holding it together. That's the cell envelope, that whole structure, okay? That consists of a cytoplasmic membrane, which all bacteria and archaea have, at least that cytoplasmic membrane. It's just holding the crap together. Cilia that's a good question, Destiny. I'm glad you asked it. Cilia is for motility for eukaryotes only. So bacteria do not have cilia. It's a good question, and it's a good one to remember that distinction. Cilia can be used for motility only for eukaryotes. Think of like whenever you think those structures that have like those little hair-like appendages that are like this, feeding in the water, but it's usually multicellular organisms that do that, or it could be unicellular, but they're complex, right? So those are associated with the eukaryotes, right? So these guys, the bacteria, they technically are true cells because they have a membrane, and we do have genetic material in there, but they're not. So the eukaryotes just have a true nucleus, and because they've already got that eukaryotes, true, U means true. So those eukaryotes have that nucleus, and that's a sign that they're going to have other structures that are membrane bound as well. So they're complicated cells. So the bacteria are way less complicated, and they're always single cell. Bacteria and archaea are always single cellular organisms. We are eukaryotes, but our E. coli in our gut, they're still alive. They're still cells, right? They're just eukaryotes. They don't have a membrane bound nucleus. In fact, they themselves are membrane bound, but nothing else in them is. That allows them to be much smaller than our cells. So they can infect our cells and get inside of our cells and multiply in them. Not all of them do that, of course, but they have to be small to do that, right? They're significantly smaller than eukaryotes when we start looking at the size difference there. Okay, so we have always a cytoplasmic membrane that contains the cytoplasm, the jelly inside the cell, and everything that's in the cell. We will have a cell wall. Some bacteria have that. We know that gram positives and gram negatives, true gram positives and negatives, should have a cell wall. It's just whether it's thick or thin. And then we have the outer membrane if you are a gram negative. So an extra membrane, if you're gram negative, if you got the thin wall, you need a little extra support. So you have that membrane on the outside, the outer membrane. To get more complicated than that, we would say there are bacteria that do not have cell walls at all. Those are the mycoplasmas. We refer to them as the tenericutes when we're categorizing them. And then we have some that on their outer membranes, or sorry, excuse me, some that are gram positive technically. Anyways, it just depends. They'll stain gram positive, but they're not true gram positives. The mycobacterium. They'll stain gram positive because that wax that's on the surface of their cells, the mycolic acid. That mycolic acid is waxy and will hold on to the crystal violet really well. But they are different. They've got mycolic acid. None of the other ones do. So that's what makes them special. So you can sort of categorize the bacteria that way as well. So whether they're gram positive, negative, or are deviants. Mycoplasma, no wall, and mycobacterium that have the mycolic acid wax on the outside. Remember, I'm going to remember that because if I look at a mycoplasma, I think of a cup of plasma, my cup of plasma, I don't know. It works for me. And if you spill your cup of plasma, does it retain a shape of any kind every time? No, right? It's just gonna go everywhere. Pleomorphic, if it's pleomorphic, it doesn't have a cell wall. Cell wall isn't there to maintain the shape, the cocci or bacilli shape, okay? Mycobacterium, if you have a my cup of bacteria, that's not really the same situation. So you can't apply the logic to that. So that you can't say, oh, that's the one without the wall, that's the one with no shape. So, no wall. So you have a cup of bacteria, no spilling it, that doesn't apply here. Okay, the plasma is what you spill. So that's the mycolic acid, mycolic acid. So that's what you should be thinking here, mycobacterium. That's how I distinct between those two. Yeah, I tell you, which one is acid fast. So acid fast staining tells you if it is going to have mycolic acid on that surface, and all mycobacterium species are acid fast, and vice versa. If it's acid fast, it has to be mycobacteria. So there's also nocardia that's another one that falls into this category, but I don't give a crap about it because it doesn't do anything medical in any way. So we're focusing on mycobacterium, and it makes it easier to just learn about one. So that mycobacterium has the mycolic acid on the outside that repels a lot of stuff because it's waxy and makes it really hardy. It's like Mycobacterium tuberculosis. It's very resistant to a lot of things. Okay. I was going with that, but yeah. We learned about the gram stain today. I'm not going to go over that one again. If you guys need to review that, I would recommend probably going through the handouts for the lab or the textbook for the lecture. So I don't want to talk about that again. I hope we've killed it. The endospores, these are dormant structures that bacteria make to withstand the harsh conditions. Usually there's no nutrients of some kind. So they make endospores, but only Bacillus and Clostridium do this. Not all bacteria. Do all bacteria make endospores? Absolutely not. Only Bacillus and Clostridium. If its name isn't Bacillus blah 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 or Clostridium blah blah blah, it doesn't make endospores. Okay. So vegetative cells are, in any case, wherever, whatever, any kind of bacteria, vegetative cells are the free living, they're multiplying, they're feeding and growing and stuff, right? Endospores are completely dormant. They do nothing. They do not move. They do nothing. They're just waiting. Okay. The sporangium is like if you're pregnant, right? The woman that's carrying the baby would be the sporangium, and the endospore would be the baby. Okay. That's exactly how I would look at it. The endospore forms inside of the sporangium. Okay. That process of making, why are you crying? Who's crying? Come here. Oh, we're all crying, apparently. All three of us. Good. I know, I'm right here. So yeah, so sporulation is whenever you're going through the process of making spores, and germination is whenever you're going back to the vegetative state. Like think of a seed germinating, like it's growing something out of it, right? You get a sprout out of it. So it's actually becoming active whenever it was kind of dormant. That's germination. So that's how you can break it down. Sporulation, it sounds like it's making spores, right? Endospores. Don't shiver so much. I don't know why you're shaking like that. But yeah, so I would be, if I were to ask you to draw the cycle of endospore formation and how it goes from like vegetative through sporulation, making the endospore via the sporangium, and then label it all the way around back up to vegetative. I'm not going to ask you to do that, obviously, because it's multiple choice, but I would recommend being able to do that so that you can be aware of the steps that are involved in endospore formation, the whole cycle. Then we have archaea, our extremophiles. Yeah, okay. Halophiles, high salt. Thermophiles, high temperature. Cycrophiles, cold temperature. We'll have acidophiles. Go figure where that would be, right? Acid. Bergey's manual is just a manual we use to identify. I'm supposed to tell you to learn how to use it in the library, but it's like a nightmare. It's like a set of like 17 books or something that like you go to a page saying, oh, I got this result on my McConkey auger. Shoot. And it's like, okay, next, refer to page, blah, 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 for, enterobacteria or something. And it's just like, okay, then you find the next thing that lines up, and then it goes to the next, it's just like another book. And then it's just, that's Bergey's manual. They have an online version of it, but we don't have access to it at OSU OKC because we're cheap. You can quote me on that. And then we have our terminology based on whether you have a cell wall or not. If you are a old cell, I say old cell. To me, that's what archaea sounds like, right? So archaea, old cell, I think of, I'm sure you guys have heard this in the lecture already. Oh God, but you're stuck in my, hold on. You're doing dumb stuff, meatball. Got stuck, his leg stuck in my headphone cord. I'll hold it up here. Little peeping sound, sad. Anyways, yeah, Joey Mendoza. He was one of my first boyfriends, and I think of that being a long time ago, right? So archaea falls into Mendoza cutes because Joey Mendoza was cute. Okay, it was a long time ago. I think I was like 12, I don't know, something like that. And then gracilicutes, firmicutes and tenericutes, the way I remember those ones. Firmicutes, firm, it's firm because it has that thick cell wall peptidoglycan, right? Then that leaves gracilicutes and tenericutes. Gracilicutes has a G, so gram negative. Tenericutes, no cell wall, there's just a leftover one. So that would be the mycoplasmas, but no cell wall. Okay. Know your terms for your shapes and your groupings and arrangements. Know the difference. Don't confuse staphylo with round cells. Staphylo doesn't mean round cells. Staphylo means, irregular bunching. It's an arrangement. Cocci means round cells. Just because it's staphylococcus, don't mix up the terms. If I were to say staphylococcus, I now ask you what is the cell shape, you should be able to tell me that it's going to be round. And I ask you the arrangement, you should be able to tell me that it is, irregular grape-like clusters. But if I ask you for the arrangement and you click cocci, that is incorrect. Cocci is a shape of the cell. So don't get them confused. That's what I'm saying. Functions of the capsule. Know that it's for sticking to things, adherence. Know that it is for helping evade the phagocytic function of immune cells. So your white blood cells want to go out and eat up. They literally like bend their whole cell around something to like eat something up and then digest it within the cell. That's called phagocytosis. We don't need to know the term right now. We're going to learn it later. But capsules help with evading that because that's sticky coating, so they can't grab on as well. And then they also help with making biofilms. So bacteria can grow on top of one another, create plaque on your teeth, for example. That's a biofilm. How do bacteria locomote, move around, motility? How are they motile? Only flagella, right? Bacteria is only flagella. Are you okay? You gonna survive it? All right, flagellar arrangements, be able, if I give you a description, apply the term to it. Cell envelopes, know the difference between gram-positive and negative and the other random ones, mycobacterium and mycoplasma. I know what the difference is. Oh shit, sorry, I wasn't expecting this. Where are you going, meatball? Stop. Stop, dude, stop, I'm serious, stop. Jeez. Gotta be the chunky one. Buddy, it's okay. It's okay, buddy. Yes. Know what makes them different. So remember gram-positive, thick peptidoglycan layer. Know that that peptidoglycan layer has lipoteichoic and teichoic acid. That just helps maintain the stability of that cell wall, especially during division. Gram-negative, it has the thin cell wall. And that thin cell wall also has on top of it the outer membrane that has lipopolysaccharide, aka exotoxin. Endotoxin. I don't know why I said exo because outside is why I thought it, but endotoxin, it's part of the cell, so endo. And mycobacterium with the mycolic acid and the mycoplasmas, they don't have a cell wall, so they are going to be pleomorphic. All right, the gram stain. Just know how the crystal violet is working, getting stuck in the peptidoglycan. The iodine is the mordant that it is going to fix everything in place. The alcohol is going to rinse out any crystal violet that didn't get stuck in the gram negatives. And then the safranin is going to counter stain. Okay. Turn this gram negatives. Negative red. Red is a negative color, right? Purple is going to be either purple or red. Crystal violet. So purple, positive. Purple, positive, PP. All right, endospores, we just went through that. Know that it's going to be Bacillus and Clostridium. Be aware of that. That's one that should stick, okay? And then categorizing based on cell envelope. That's all that tenericute crap that we just went over. Mendoza cute, tenericute. What are you doing? This is not bed. All right. The eukaryotes, we have a little bit less going on here. Remember, okay, so either one can move with flagella. Where are you going? I'm not kidding. Where are you going? Flagella and cilia, are how our eukaryotes are going to move. They can also move via pseudopods. So it's just sticking out part of the cell, like, and then pulling themselves along. Those are pseudopods, or pseudo being fake, podia feet. Okay. Yeah, so the flagella in eukaryotes are more complicated. They have a membrane surrounding them. Remember, in bacteria, the flagella are going to rotate like a motor. In eukaryotes, they're going to whip. And they also have microtubules holding them in structure in the middle of it. So glycocalyx in multicellular cells, the glycocalyx is going to form the extracellular matrix, not like a capsule or something. If it's unicellular, it can still have capsule-like structures. Organelle is anything that is membrane bound. That includes the nucleus. So these are eukaryotes, true nucleus. So the nucleus is membrane bound, and there's other things that can be membrane bound. So mitochondria create the energy, endoplasmic reticulum, basically protein production and other molecules in the smooth portion. The Golgi apparatus is involved in packaging and vesicle formation. Chloroplasts are for photosynthesis. And then the nucleus is the genetic material. And it's where transcription is going to happen. We have mitosis. Mitosis is whenever cells divide to create exact copy daughter cells. And that's it. So like if you scrape your knee, you go through mitosis to heal that. You're not going through meiosis. Meiosis creates sex cells. So it's a different process. There's more steps. Mitosis, just making exact copies. The steps for mitosis, PMAT. Puppy pees on the mat, okay? Prophase, you're gonna be just starting out. Everything's just getting ready, condensing everything. Metaphase, lining them up in the middle. Metaphase, middle. Anaphase, you're gonna start pulling apart. Anaphase, apart. And telophase, you're gonna make the new membranes and stuff with, pinching off those daughter cells. So that's the steps. Really kind of all you need to know about mitosis, okay? Right, and the fungi, these are terms here for the fungi. This is what I hate about the fungi. Be familiar with the terms for the fungi and be able to tell them from one another. If I tell you, what is the open, a spore forming structure for fungi that doesn't have a covering over it, and I give you choices between conidiophore and hyphae and sporangiophore and whatever, another like mycelium, then you need to be able to know the difference between all of those. That's basically what you need to know about the fungi. There's another term in here, saprobes. That's one to be familiar with. It will come up a little bit here and there. Just saprobes are organisms that break down the dead things. Protista is a term for the algae and the protozoa. The algae are going to consist of a lot of our oxygen creating organisms in the world, like our phytoplankton and stuff like that. And it also includes like sea kelp. So they can create, they're not true multicellular structures, but they are like organized cellular structures, okay? The protozoa move either by pseudopods, that's what amoebas do. Those are protozoans. Flagella, or they can have cilia, or they don't move at all. The ones that don't move at all are all parasites. That means they're going to all be feeding off of other creatures somehow, usually disease-causing parasites. So like malaria falls into this category. That's the apicomplexans. Protozoans can also form structures that are resistant. They're dormant and all that, like endospores, but they're called cysts. So if it's a cyst, think of it as being all balled up, like a cyst would be endospores inside of itself. So the cysts are like that. You've got to be out of your mind, buddy. I don't know what you think is happening here, but I hate it. I gotta be honest with you. Anyways. I hate it so much. Yes, so the cysts, yes. Trophozoite is a nice fancy word, but think of troph like a trough, like pigs eat from troughs, right? So if you're eating from a trough, you're active. So you're not dormant. So you gotta be, moving to eat. Remember that cysts do nothing at all, not even eat. So that's how I tell the difference. And as I remember that endospores are like cysts, Trophozoites are like vegetative bacterial cells, okay? Then we have the Animalia group. Yes, the helminths, the worms, they all fall into Animalia. They're all multicellular. And they have their own nice little words. So helminth, and then for the roundworms, we have nematode, flatworms, platyhelminthes, flukes, trematodes. And do we have more for cestodes for the tapeworms? Those are flatworms, obviously. But flukes and those also are kind of broken down. So we'll break it down into categories. Are you roundworm or are you flatworm? And if you're flatworm, are you fluke or are you tapeworm? Okay. So what does it look like? Okay, let's do for multicellular eukaryotes, extracellular matrix, kind of what holds your cells and your organs and stuff together. An organelle is a membrane-bound functional unit in a cell, like the mitochondria. 5 major organelles and their functions, we kind of went over those. Basic understanding of mitosis, not just PMAT, but what is mitosis for? So creating identical daughter cells, right? Not for sex cells. That's meiosis. Fungi, be able to discern between those terms. If I slapped them all up there and you have to choose the right one for a definition, and they're all fungi related, know the difference, okay? Protozoan, dormant versus active states. That's going to be the dormant is the cyst, active is going to be the trophozoite. That's analogous to bacteria. Dormant is the endospore and active is the vegetative. And the helminth terms, which we kind of went over just a second ago. Helminths are either going to be roundworms or flatworms. Roundworms are called nematodes. Flatworms are called platyhelminthes. Platyhelminthes, you have flatworms, you have flukes, or you have tapeworms. The flukes are the trematodes and the tapeworms are the cestodes. Okay, next. Chapter 7, viruses. Viruses are obligate intracellular parasites, and they're simple, non-cellular, and lack organelles. So these guys are not cells. They are acellular, without cells. They are not alive. They are not organisms. They are viruses. They're obligate intracellular parasites, meaning they cannot survive outside of a cell, okay? A single virus, most of my graduate work was on viruses, by the way, so I'm pretty well-versed on viruses, but that sometimes means that I think that it makes sense to people when it doesn't. So, you know how that gets, like when you know about something, it's not on purpose. I don't do it on purpose, just FYI. So yeah, so taking advantage of, that's not what I'm trying to mean. Okay, forget it. A single virion consists of just a genome at least, right? And a capsid at least. So there's 2 things that you absolutely have to have: genome, capsid. The genome can be any kind of nucleic acid. It could be single-stranded, it can be double-stranded, it can have positive sense, which means you can just read it right away how it is. It could be negative sense, meaning that it has to be, you know, made into its copy in order to be read, which we'll learn more about that in genetics. Sometimes things throw me off. I'm so sorry. I have ADHD and it doesn't serve me right sometimes. Well, most of the time it doesn't serve me right. All right, but yeah, genome can be anything of nucleic acid form. RNA, DNA, single strand, double strand, positive, negative, doesn't matter. Capsid is the protein coding. So the genome of your virus has to be coded somehow. What are you doing? Sitting on like my chest. So these dogs are weird, man. Anyways, capsid is the protective coating. It's going to basically just protect. You can't just have nucleic acid necessarily just floating around. So you will have a covering on it called the capsid. You have to have those 2 things every time. 100% of viruses have those, okay? Spikes that help the virus get into cells, they're called, they're made of glycoproteins, so sugar and protein mix, just like act like a key to get into the door on your cells. Some viruses also have an envelope, which it steals from the host that it came from. So when you have COVID, the COVID little capsids with its genome pushes its way out of your cells because it's inside of your cells. pushes its way out and covers itself with a blanket of your cell membrane and carries it around like a, freaking coat, basically. And that is its envelope. It can even stud the envelope with its own proteins ahead of time so that it can still have its spikes on the outside. Viral life cycle: absorption, penetration, uncoating, synthesis, assembly, and release. 1, 2, 3, 4, 5, 6. It's supposed to be 6. Assembly and release are separate from one another. No, if I asked you to tell the story of the virus, don't just tell me these 6 words. Tell me what each word means and how they relate to one another in terms of the virus, right? Cytopathic effects are negative changes in the cells caused by a virus. Could be a whole lot of things: not sticking to surfaces anymore, rounding up, merging with cells nearby, all sorts of things. Anything bad change-wise from the virus. Bacteriophages are viruses that only infect bacteria. They infect only one species of bacteria. So a T4 phage only infects E. coli. It can't infect any other kind of bacteria. But E. coli can be infected by a whole lot of different kinds of phages. All of those only infect E. coli. So it's kind of neat how that works. But they inject their DNA into the bacteria, the phages do, and then the bacteria make copies of the phages and they just burst out of the cells, called lysis. That's how they get out. In human viral infections, we have budding out, so pushing out through your membrane, like coating yourself like a little coat around your little viral body and taking it with you, or exocytosis, where the cell actually pushes you out actively. So like as if it was manufacturing you and then making something that it secretes out. We do that with the viruses. But yeah, bacteriophages always burst out. Lysogeny, this is whenever a temperate, that's a term also you should know, temperate phages can undergo lysogeny. Lysogeny is when the temperate phage incorporates its genome into the bacterial genome. This is known as then a prophage when that happens. And that prophage can be multiplied out whenever the bacteria multiplies. It carries it to the offspring. And then if it goes back to making viruses and busting out again, that's the lytic phase. So it'll cycle between those. Temperate phages can cycle between lysogeny and lytic phase. The provirus, people look at the camera. Good boy. Lysogeny, lytic. Yes, I'm happy with those. That's fine. You can also have something called lysogenic conversion. So while you have your prophage incorporated into your bacterial genome, it can have other genes that are beneficial to the bacteria, like making toxins or antibiotic resistance or something like that. That's lysogenic conversion. Anytime, and most, I'm not going to lie to you, most bacteria that are pathogens, a lot of them got their traits from that, from lysogenic conversion. So it's pretty neat to think about, especially when I tell you that a very large portion of your genome consists of viral DNA from just inherited, that we don't know what it does. So think about that. So a provirus, this is where we get back to the eukaryotes getting infected, like you and me. A provirus is whenever you have a virus that incorporates itself into your DNA. There are plenty of viruses that do this, like HIV or herpes simplex virus, okay? So it's not a weird thing. Chickenpox does it. Any herpes virus, chickenpox is a herpes virus. So it'll incorporate itself into your genome, and it happens with HPV as well. When that happens, it can incorporate itself sometimes into random spots in your genome, and that can lead, or in specific spots that can be bad for your like mitosis regulation or your cell cycle in general. So that can make things in your cell cycle go completely off kilter and lead to over replication of your cells. That's cancer. And those viruses that can do that are called oncoviruses. Hep C is one of them that can do that, and so can HPV, human papillomavirus, that causes cervical cancer. It's the absolute #1 most common sexually transmitted infection out there. Now, about 90% of people who are sexually active will have had it by the end of their lifetime. I'm not gonna lie, I've had it before. I tested positive for it, Because the only reason I know is because I go to my pap smears. If you're not going to your pap smears, you need to be going to your pap smears, because that's how you get rid of cervical cancer before you get it. And I had one of those strains, and I had cells removed. I was treated for it before it became cancer, and that's how it should be for everybody. I'm going on living a normal life and don't have the virus anymore. My body kicked it. All right. Prions are just protein. You remember how we we're talking about amino acid ribbons and how they'll fold back on each other, and how, like, oh, they'll fold back like this, and it's not happy, and then they do a big folding, and then they interact with other parts, and they're functional. Imagine if you were happy and you're like this, and one that has the exact same ribbon, green ribbon and green ribbon, okay, comes along and it's like this, and it's like, hey, why are you doing it like that? Why don't you be like this? And then the other one is like, oh, you're doing it like that? Well, I'm green ribbon does this. And it's like, well, I'm not. That's what prions do. Okay, that's how we think they work, that we have misfolded protein that will come along and just train your regular good protein to not fold the way that it's supposed to. If it's not folded the way it's supposed to, it doesn't work the way it's supposed to. And if it is folded like this, it might have had stuff in here that was hydrophobic, that it was hiding away from the environment around it, you know, on purpose. But now it's out like this. And so what happens is you have a lot of hydrophobic groups that will be out there, don't want to be out in your cells and in your body and the water, basically. So they'll just stick together. And they'll create these to hide from the watery environment. That's basically called amyloids or plaques, and they build up and create holes in your brain, and that's what causes spongiform encephalopathies. So encephalopathy, encephalo means brain, pathy means something's wrong with the brain. That's all that means. Spongy means literally sponge, and form looks like a sponge. It's form of a sponge. Okay, not as complicated as it looks. All right, so spongy form, sponge-like. Encephalopathy, something wrong with the brain. That's all that means. Okay, so they're just proteins. They do not have a genome of any kind. Okay, so all viruses have a capsid. Not all of them have an envelope. All viruses have a capsid. Not all of them have an envelope. Okay, can't say it enough. The capsid is the protein that covers the genome, keeps it safe. The envelope is a membrane that it may or may not have. Not all of them need it. Parvovirus, for example, doesn't have a membrane. It's one of the smallest viruses that we know. All right. Why wouldn't bacteriophages have envelopes? They are busting out of the cell. They're not, budding out nicely. So they don't, and they're just going to inject their stuff in anyways. They don't have to merge with the host. So a lot of our viruses will be membrane covered because they'll come up and just dock with our cells and then fuse the membrane and just empty their little contents into your cell so they can go do their thing. And it's how viruses work. Isn't it lovely? I'd be aware of the basic parts of most viruses. So not just the genome and the capsid, that's the only necessary ones. Be aware of that some of them have an envelope. Be aware of what spikes are and what they can do. All right. And what kinds of genomes they can have. Virus life cycle and what the terms mean for what's happening with the virions. That's those 6 steps that we were just looking at, the absorption and all of that. Latency versus lysogeny, that's going to be human versus bacterial, literally how that's organized, and that's how the second one is as well. Latency is like when you get the chicken pox, and then way later, decades later, you get shingles. Okay, it hid out in your DNA. And you're a human being, you're a eukaryote. In bacteria, when that happens, it hides out in the DNA like lysogenic conversion, and it comes out again, it will be switching into the lytic cycle. Provirus is whenever you are a human being. Do you need to jump down? I really wish you would. Provirus is whenever you are like eukaryotes and you have your genome of the virus incorporated into you. And then the prophage is your bacteria, same situation. Lytic versus lysogenic. Lytic is the active replicating viral form of the phages, and lysogenic is going to be when the prophage is hiding out in the bacterial genome. be aware of which terms apply to eukaryotes versus bacteria, okay? Don't get confused from them. Role of lysogenic conversion in disease is actually pretty common. Like I said, we just talked about that. How could viruses cause cancer? We talked about that. Could cause disruption in the cell cycle, and depending on where it inserts itself into your genes. And then a genome of prions, they don't have one. Just kind of a trick little statement there, FYI. And what diseases they cause. I say diseases that spongiform encephalopathies. It's a group of diseases. It's a type of like group. So there are diseases that fall into that. You don't need to necessarily know the different diseases. But for example, it's going to be mad cow disease, Creutzfeldt-Jakob, and the variant form, which is the human form of mad cow disease. Fatal familial insomnia, where you die from not having enough sleep. And then Kuru, it's cannibals. Literally, that's what it is. People eat people and then they get the disease. And then chronic wasting disease in deer and scrapie in sheep, which is the first one that was studied. Well, those are just examples if you're interested. But yeah. Okay, so that is all of that information in the review for that. That took me a lot longer. I know that you guys just haven't had a lot of attention from me for a while, so I'm not going to play the Kahoot today. I'm going to post the link to it on the announcement. Okay, so once this video is done and I can upload it, then I will post a link to the video, post a link to this slideshow in case you want it. And then I will post the link to the Kahoot as well, so you can play through it tonight on your own free time, because this went way longer than I wanted it to. So do you guys have any questions?
No, ma'am. Thank you.
Awesome. Anybody else? Anybody else? All right, so I'm going to go ahead and close out of this. If you guys think of anything later while you're studying, please email me. Remember, I mean, we start at 10 a.m. Some of you guys start at 11:30, depending on what class you're in. But when we start, that's how long you have to study. So you can always email me before that, and I'll try to answer your emails as best I can. Anyway, I'll see you guys tomorrow for the exam. Remember to bring your laptops. If you don't have lockdown browser or you're confused about it, we can always do it whenever you get to class. I just don't want everybody doing that. And it will be in the lab. So if you want to clean off the countertop before you get started, you can, and you do not need to wear your lab coats. Okay. And I'll remind you when we get there, everybody's gonna want to try to, it's okay. But you don't have to. If you want to, be my guest. All right, that's it. Okay, meatball, cheese.
Thank you for the review. I appreciate it.
Yeah, you're welcome. I don't mind. Yeah. All right, I'll see you guys later then. Get some rest. Happy studying.
Actually, Professor Yost, could you say a little bit more about the virus synthesis? Because I know you said that they have to have, once they unravel their capsid and the DNA or RNA actually gets transmitted into the cytosol of the recipient cell, is it that if it's a positive sense strand, it can just be used to make new proteins? But if it's a negative sense strand, then it needs to undergo, is it transcription?
Yeah, so yes. So making a copy or replication, I guess you could call it. It's a form of replication. But yeah, so if you are a positive sense of whatever, especially RNA, right? So we know RNA is, well, we're going to learn about it in the next chapter, but RNA is our messenger molecule. It's our in-between. We have DNA as like our blueprint in our Smithsonian. We can't take it out of the nucleus. And so we go in there and we pick to make a copy of our gene that we want to make a copy of. So we make that copy in the form of RNA. So that's our working copy. So that's transcription. So DNA to RNA is transcription. And then you can take the RNA into the cytoplasm where translation will occur, typically in the endoplasmic reticulum. So that is, thanks to the ribosomes, that is the central dogma. So DNA, RNA, and protein. DNA to RNA is transcription, RNA to protein is translation. Anyways, with viruses, if they have RNA genome, a positive sense RNA genome, like we were talking about, that means that they can just pop a ribosome on there and start making protein right away on that genome. If it's negative sense, it just means they have to make a replication copy of that negative strand. So if it's negative RNA, make the positive RNA, and they can use the positive RNA to then read and make protein and protein and protein and protein. With the DNA viruses, they have to move into the nucleus. And then in the nucleus, they will go through the normal process of DNA to RNA to protein. If they are single-stranded, then that's just, they don't have to split the strand up. They just sort of one strand, and then they either make a copy or they make a copy and then a copy, depending on if they're positive or negative. I'm not going to have to worry you guys too much about telling the difference of like literally breaking it down between how positive and negative and DNA and RNA work for the virus. I won't necessarily ask you about that. I would be aware that the DNA viruses have to have a replication step in the nucleus. I would be aware that RNA viruses can do that in the cytoplasm. They don't need to go in the nucleus. And that, because we haven't really learned about, transcription and translation yet. So I don't want to get into too much detail with that for the viruses yet. Does that make sense?
Yes, it does.
Does that answer your question too? I hope it did. I know it was a lot.
Yeah, the one of the charts I think on the textbook kind of just confused me because it mentioned like what is it, RNA polymerase and the
Yes,
replicase.
So some viruses like HIV has a RNA genome. And so obviously you'd think, okay, RNA, like it's just ready to go. Are you serious? It's just ready to go. Sorry, these dogs, man, I swear to God. Ready to go with being, make protein and oh, it's happy and whatever. What DNA does is it wants to insert its genes into your genes so it can hang out there and you'll keep it safe, right? So to get RNA to go into DNA, that's the opposite of everything we've ever learned. So DNA, RNA, protein, right? That's usually how it goes. Well, what HIV does, it takes this RNA genome and it turns it into DNA in a process called reverse transcription using reverse transcriptase. So it brings a special new enzyme along basically for the ride so that, and it codes that in its own genes so that it can do that. And so it's very unique. And then it will incorporate that DNA that it makes into your DNA. And then it will use that to make more RNA and make more viruses that way. So that's how HIV works. Reverse transcriptase, I'd be kind of aware of the general concept maybe for now, but don't worry too much about the details for now. I'm not necessarily going to ask you about the life details. Be aware that HIV has reverse transcriptase. Maybe that's about it though. I don't think I'm going to ask you about that. If it's not on the sheet, then I probably won't ask you about it. Because I go straight from that sheet, I'm not going to lie. There's not direct questions from there, concepts that I pull from there, but like if it doesn't mention reverse transcriptase on the virus section, then I'm not going to mention it in the test.
For the peptidoglycan, it's the cell, but it's the outer, it's the cell wall though, right?
Yeah, it's like it is exactly what it sounds like a wall surrounding the cell.
It's a wall surrounding the cell. Okay.
Yes, and it gives the cells their shapes.
Okay.
And it kind of helps them with like, if it's too salty of an environment or too, not enough salty.
Okay.
We'll learn about that with osmosis and all that. But yeah, it'll protect from that.
Okay, that'll work. Thank you.
Yeah.
Are the was it lipoteoic acid and then the teoic acid of the shoot. You know what I'm talking about. Are those considered are those considered cholesterols?
I don't know enough about them and how their structures are. As far as I know, that they are not considered cholesterols. They have a lipid quality, obviously, lipotecoic, lipo. So a little bit waxiness to them. I believe they're amphipathic molecules. I won't ask you guys about that. I will say eukaryotic cells, our cells don't have cell walls, but they can have kind of structure and they need to maintain kind of shape. You know the difference. Sometimes whenever you look in a microscope between muscle cell versus a skin cell or something like that. So they maintain their shape by cholesterol. So our cell membranes are actually rigid because of cholesterol. So good question. They might be, I think it might be a molecule related to cholesterol, but it's different still, but yeah. I won't ask you about it either. But these are good questions. I like it anyway. Stuff I don't really know about.
Do you mind briefly going over the difference between temperate phages and prophages?
So a temperate phage is just a type of virus, basically a bacteriophage, that can undergo lysogeny, or switch between lysogeny and lytic, right? So when you are switching between those two, lytic is when you're making your copies and bursting out of the cell for bacteria. Or if you are lysogeny, you will incorporate your whole viral genome into the bacterial genome. When you do that, the actual incorporated bacterial genome, the actual incorporated viral genome into the bacterial genome, the viral portion is called a prophage when it is incorporated in there. So temperate phages can undergo lysogeny. When lysogeny happens, and it's actually incorporated in there, then it's called the prophage when it's in there.
And both of them can be reactivated into the lytic phase. Is that right?
So yes, they're the same. So it's like kind of the same thing. It's like a temperate phage is just, I don't know how to describe it. Like if I were to say T4 phage that infects E. coli is a temperate phage. Then T4 can infect E. coli and go through lytic phase. That T4, which is temperate, can go through lytic phase and just multiply and burst out. And that's that. Or because it's temperate, it could incorporate its genome into the E. coli genome. And then when it is incorporated into the E. coli genome, each time the E. coli will like divide, the bacteria itself divides, it's also dividing with the viral genome in it, right? And no matter how it got that, either from the division or from the initial infection, where it's just copied from dividing and dividing and dividing, that incorporated viral DNA is called the prophage. And at any point, that prophage could pop out and go through the lytic cycle and then, start making viruses and pop out again. But that whole category of a virus that can do switch between is called a temperate phage. Just the most absurd thing. I don't know what your deal is, man. He literally never sits on my lap. I don't know what this is. It was weird, I guess.
So lysogenic conversion occurs whenever the temperate phage infects the host cell, not when it becomes not when it becomes a prophage.
So both. So both. So if a temperate phage infects a host cell, it can become a prophage. And when it becomes a prophage, that is lysogeny. So lysogenic conversion happens when lysogeny has happened and that incorporated prophage also it includes with it genes for toxins or whatever will help the bacteria be more pathogenic, cause more disease. So temperate phages, when they undergo lysogeny, they are incorporating their genome into the bacterial genome in a form called the prophage. Prophage can carry with it genes that can help the bacteria be disease causing. And that is called lysogenic conversion. And then it could pop back out again and go through lytic phase. At any point. Dude, you have got to be kidding me with this. Over it. You're too hot. Okay, if you guys, are slowed down on your questions at least. I'm gonna go ahead and cut off here, and if you guys have any more questions, you can always email me and I'll try to get to them as soon as I can. So I'm gonna go now. I'm just trying to figure out what these weird dogs need. I'll talk to you guys later.
Thank you.
Yep.